So we mentioned that the first pathway for any type of cellular respiration is glycolysis. Number one. First. And it is going to be the breakdown of sugar to release energy. It doesn't require any oxygen. And so it makes it anaerobic in nature. It is going to result in the net production of 2 ATP that can be used for the cell for cellular activity. And it's going to leave behind 2 pyruvic acid, like we mentioned in our first flow chart. So glycolysis happens in the cytoplasm of a cell. All cells have cytoplasm. All cells can go through glycolysis. And it's going to start with our C6H12O6 molecule, represented on your diagram with a kind of hexagon-ish shape, right? Each one of these representing a carbon, and the hydrogens and oxygens aren't represented in this diagram. The first part of this is going to be an investment in cleavage. And what's happening in the investment in cleavage phase here is that energy is going to be used here. We'll invest some energy in the form of ATP, and that produces our byproduct of ADP. And it's going to result in the production of a molecule you already recognize as G3P. And if you remember, in the Calvin cycle, we had produced G3P, and that could be used to put 2 of those together to create glucose. So now we're going to be creating 2 of these G3Ps. And so I'm going to represent them with our 3 carbons together. There's one of them, and there's our other G3P. So our first part of glycolysis results in the lysis or breaking of glucose, hence the name glycolysis, and it results in our G3Ps, which do have a phosphate on the end of them. All right, that phosphate wasn't there up here in glucose. Glucose is C6H12O6, no phosphate. However, with the addition of ATP and some energy investment, we now have phosphorylated and cleaved our sugar, and we now have our G3Ps. Now, these G3Ps have plenty of energy stored in them, and that energy is going to be released or removed in a series of reactions that happens on each of these G3Ps. Each of those is going to go through an oxidation in which NAD positive, sounds familiar, is going to be turned into NADH. And in this case, you'll notice there's no P. In photosynthesis, we had NADPH. Now we have an electron carrier that is without the P in the name. We're also going to have the production of 2 ATP, and that's going to be 2 ADP that are turned into 2 ATP. And that same set of processes is going to happen over here to the other G3P. So by the end of glycolysis, we'll have made electron carriers and we will have made ATP. And these 2 ATP are useful. These are our molecules of energy for use by the cell. And we are left with 2 molecules of pyruvate. And pyruvate is a 3-carbon molecule, also known as pyruvic acid. And each of these pyruvates or pyruvic acids must be dealt with. And there is a lot of energy left in these molecules. And that what's going to happen to those molecules is going to depend on whether or not there's oxygen available to the cell and whether or not there's the correct cell machinery to deal with it. So let's just do our accounting here at the end, accounting of our glycolysis process. The accounting is that we invested 2 ATP, right? So that's a negative. But then we made 4 ATP. So we are left over with 2 ATP that can be used by the cell. So these ATP can go off and be used by the cell. We are also left with 4 NADH, which have energy left to them, but they are not by themselves going to be the currency of energy. They're not going to be usable energy. And we also have 2 pyruvate, and the pyruvate actually are packed with a lot of stored energy still. So these molecules right here, depending on whether we're going to go down an aerobic path or an anaerobic path, it changes where they're going to go.